Hello everyone, welcome back to Tamali's classes. After having a lot of thought about what to talk to you about in this video, I have decided to go on with this year's Nobel Prize winners and also the topic that was selected for Nobel Prize in Physiology and in Medicine. So, topic here this time is the discovery of peripheral immune tolerance. So, have you heard about this topic before? Well, it is a topic that is related to immunity, and three scientists, Shimon Sakaguchi, Mary E. Brunko, and Fred Ramsdell, they have worked on this peripheral immune tolerance. They have discovered it, and that led to Nobel Prize winning in physiology and medicine. So, what is this topic? Let's go into the depth, and I'm sure you love it. Watch till the end, and do not forget to subscribe to the channel, Tamali's classes, and also share this video with your friends. So, let's start. Before we go on to their work, let's talk about a bit on the basics and introduction to immunity. Well, in my video classes, I've already spoken about the immunity and I've already introduced the immunity part. So, if you want to know more about it, you can go and watch my previous classes on immunity. Today, I'll just touch the basics out here. So, first, we'll talk about the types of immunity. So, there are two types, innate immunity and adaptive. Innate means this immunity which you are born with. That means when our body, when they are coming across some certain foreign microbes or foreign particles in the body, how they react to it. That is the first line of defense. So that is the innate immunity. And other immunity is the adaptive immunity that develops throughout your life. That means it is basically antigen specific, like it will uh, identify the antigens, the foreign bodies, and it will directly go and kill it. So out here, even antibodies are produced. So this I've already discussed deeply in my immunity classes. So please go to the class if you want to know more about it. So this is innate and adaptive immunity. So adaptive immunity is the later stage immunity. Okay. And also we'll talk about the B cells and T cells, which are very, very important because they are responsible for the immune responses. So B cells out here, it is produced in the bone marrow. Both B cells and T cells are produced in bone marrow, but B cells they are produced in bone marrow and they mature in bone marrow. But T cells they are produced in bone marrow, but then they move on to the thymus. Okay, so B cells they are producing antibodies which help in acting against the antigens. And T cells out here, they have another different role. They directly kill the infected cells and microbes with the help of different type of T cells. So basically, T cells are of four types. So here I've mentioned only three types: helper T cell, cytotoxic T cell, T regulatory cell, and memory cells. Memory T cells. Okay. So we'll talk about it in details in my next slide. But the name you can understand helper T cell means it helps or it monitors the immune responses. Cytotoxic T cells is which directly kills the infected cells, and T regulatory cells are different type of cells which suppress the immune responses. So we'll talk about it in our future slides. So first, we'll talk about the regulatory T cells. So it was discovered by Shimon Sakaguchi and also the other scientists like Mary Branco and Fred Ramsdell. They have also contributed to with him in finding these regulatory T cells. So what happened? Let's see. So they won the Nobel Prize in 2025, and they worked on this peripheral immune tolerance. So which includes the discovery of this T regulatory cells, which are also known as the security guards of the immune system. So out here, CD25 receptor or protein, it is a surface marker which identifies the regulatory T cells. That means if we look into the helper T cells, it has got CD4 plus, and cytotoxic T cells have got CD8 plus. But this T regulatory cells, along with CD4, it also has got CD25. So CD25 is basically the key surface marker for this T regulatory cells. Now, what are T cells? T cells I've already told you. The T cells they are produced in the bone marrow, but they are maturing in the thymus. T cells types I've already told you. So what does cytotoxic T cells do? They produce the toxic agents and they go and kill the targets. They kill the infectious agents. Okay. Now B cells, what they will do? They will stimulate. Just helper T cells. They will help regulating the immune response and also they will help the B cells to produce the antibodies. There are two functions of this helper T cell. Okay, and they will also help the cytotoxic T cells to become active. Regulatory T cells are the one which suppresses the immune responses. That means they will regulate it. Okay. So now we'll talk about the regulatory T cells because this is the main T cells that is in talk out here. So this T regulatory T cells are also known as the TREGs. They will suppress the immune responses to prevent damage to the body's own cells. Sometimes, like T cells, we know that T cells they will kill the infectious agents, or they will kill the infectious microbes or the harmful microbes. So how does it identify which is the cell's own, which is the body's own cells, or which is the foreign and body cells? How it does it identify? So out here, regulatory T cells they will suppress those immune responses so that no damage is being done to the body's own cells. So they will suppress it and it will they will prevent it from becoming overactive. And also, it will help to prevent autoimmune diseases because in that type of diseases, what happens? The body attacks its own tissues. So how does T cell discover a virus? Suppose there's a infected cell which is holding the virus. So what happens out here? They use the HLA protein complex. So HLA is the human leukocyte antigen. It is a system of cell surface proteins that are critical for immune function. That means what? These cells they help the immune system to distinguish between the self and the non-self cells to fight the infections. So they will help the T cells to identify between the self and the non-self cells. Okay, that's why it's very important. So all the T cells they have their own unique shape receptors. What they will do? These receptors, when a T cell receptor will attach to a virus fragment, the T cell gets activated, and this will alert the other immune cells to come to know that the body is under attack. That means the T cell receptors they will identify the virus fragments, they will go and bind to it, and it will alert the entire body immune system that there is a foreign particle inside the body. Okay, now we'll come to central T cell tolerance. So out what happens in immature T cells are produced from the bone marrow. Okay, so they will move to thymus for maturation. Before they move into the maturation part, these immature T cells they have to undergo a test known as central T tolerance test. What happens out here? They will bind to the self MHC molecules. So again, there are out here three steps out here, three cases. With non-selection cases, positive selection and the negative selection. In non-selection, there is no interaction between the immature T cells and the self-MHC molecules, therefore which results to the apoptosis or cell death. In positive selection, there is a binding between the immature T cells and the self-MHC molecules. So when they bind, they can easily identify which is self and non-self, and therefore the T cells survive. In negative selection, T cell binds very tightly with the self-MHC molecules. There's a strong affinity which leads to the apoptosis or cell death. Now this is the picture of the central T terminal. Just look at it out here. In negative selection out here, it leads to deletion. Okay, so they will the immature T cells specific and for the self antigen they will bind, and it will lead to the negative selection, or you can say deletion. So after that only they move on to the periphery. The ones which are not deleted, they move on to the periphery, and there again another peripheral tolerance comes into uh, picture that we'll talk about in the next slide. Okay, so that time, so before moving into the periphery, there is a regulatory T cells. Okay, it develops the regulatory T cells, which then moves on to the periphery, and there what happens? The peripheral tolerance happens. That means this is the immune system. It is a process in the immune system wherein that the mature lymphocytes they are prevented from attacking the body's own tissues, and this happens after they have left the primary lymphoid organs or after they have left the thymus. That means outside the thymus. Okay, this type of tolerance occurs where the T lymphocytes they are prevented from attacking the its own body's tissues. Okay, so it acts as a fail-safe mechanism to central T tolerance, and it uses mechanisms like by separation that is called T-Rex's Tregs. Okay, again Tregs. What happens? Regulatory T cells they suppress the immune responses. Number one. Number two, energy. Energy means the induction of unresponsiveness. Number three is the elimination of the self-reactive cells from the periphery. Okay, so by these three mechanisms, what happens? Peripheral tolerance occurs. This is very important because to um, it helps to prevent the autoimmune diseases. So when this tolerance fails, then autoimmune diseases develop. But when it is successful, then what happens? Autoimmune diseases are prevented. What do you know what is autoimmune diseases? So autoimmune diseases are those diseases in where the body's antibodies are attacking its own tissues, its own cells. That's called autoimmune disease. Okay. Now, let's see what the experiment that inspired Sakaguchi. So what did Sakaguchi do? He took a 3-day-old mice, and from that mice, he removed the thymus. When thymus is removed, what happens? The mice develops autoimmune diseases. Okay, because it doesn't have the thymus to protect it. And then what happened? After a few days, okay, after a few days from a healthy donor, mature T cells were extracted, and it was injected into those mice. Okay, so what they saw that when they injected the mice with the mature T cells from another mice, they saw that those mice were protected from the autoimmune diseases. That means it showed that something is there which is protecting the mice from autoimmune diseases. Then Sakaguchi, by this method, he defined a new class of T cells known as regulatory T cells. Okay, so he saw that other than uh, this T cells, there might be some other cells which is also carrying this CD4. So he found out that other than CD4, there's another protein called CD25. Okay, so he saw that this CD25 it was also present, and that CD25 belonged to the regulatory T cells. Okay, so this he found out by doing a test of hypothesis in 1995. Okay, so Mary Branco and Fred Ramsdell, they also worked on, after a few years, they also worked on this mice thing, and they also found that the results were related to Sakaguchi's work. Okay, what happened was they searched for this scurvy mice mutant gene. What did they do? In that, they were trying to find which was the gene responsible for the mutation that occurred in the scurvy mice. Okay, so they named it as scurvy mutation. So they saw that it was X-linked gene, okay? That gene was X-linked gene. So they did a lot of studies continuously, they did some experiments and all. And after doing several experiments, they found out that they reduced, like, there are so many genes, okay? Entire genome they studied, and then they saw that they came down to the, narrowed it down to the 5 lakh base pairs. And after that, they again kept on studying gene by gene, and then narrowed down to the 20 genes. Then from 20 genes, they studied it individually. Okay, they studied the 20 genes individually. Okay, and that led to the locating of a gene known as FOXP3 gene. Okay, they came to locate the FOXP3 gene, which they found that it was uh, this FOXP3 gene was responsible for this selfie mutation, and this FOXP3 gene it was responsible for development of the regulatory T cells also. Okay, so now let's see the series of events in details. So I've already told you about the 5 lakh nucleotides. Was then they narrowed it down to 20 potential genes after mapping the X chromosome, and then they compared these genes between the scurvy mice and also between the healthy mice. Okay, between the scurvy mice and healthy mice, and then after comparing, they found the scurvy mutation, and this faulty gene it had a lot of similarities with another gene known as the fork head box or fox gene. Okay, so these fox genes they regulate the activity of other genes which can affect the cell development. So they named it as FOXP3 gene. So this FOXP3 gene, when there is a harmful mutation in this gene, it leads to a rare autoimmune disease known as the IPEX. Okay, this is also again linked to the X chromosome. So in 2001, in Nature Genetics, they have revealed that when there is a mutation in FOXP3 gene, it leads to a human disease called IPEX and also the scurvy mice's ill health. Okay, so they understood through these experiments that FOXP3 gene was important for the regulatory T cells, which was discovered by Takaguchi. So these were all linked together. So again, 2 years later, Simon Sakaguchi and other researchers again, they proved that this FOXP3 gene controls the development of regulatory T cells. So these cells will prevent the other T cells from mistakenly attacking the body's own tissue. That means what? The T regulatory cells are suppressing the immune responses, which is again very important for a process which we have already studied, that is the peripheral immune tolerance. Okay, so this is a just diagram how the T cells are protecting us. You can see that. Okay, so this is all about your peripheral T tolerance, peripheral T tolerance, peripheral tolerance, and how it is preventing the autoimmune diseases in human body. What is the prospects of these findings? The prospects are that after they saw that the functions of T-Rex, they thought that this could be further used for numerical treatments. It could be used for other autoimmune diseases also. Okay, so they thought that if we develop more, if we form more regulatory T cells, then it will help in suppressing the autoimmune diseases. Okay, so one they have seen in regarding interleukin 2, so they are giving patients interleukin 2. This substance it makes the regulatory T cells. Okay, like it helps in the growth of those regulatory T cells. So scientists are saying that if they inject this interleukin 2, how does it help in suppressing the autoimmune diseases? Okay, they are seeing how can it prevent the organs from being rejected after transplantation, because after transplantation what happens, sometimes the organs get rejected. So how can they prevent that? Also, they are seeing that how can they slow down an overactive immune system by isolating the T-Rex from the patient and then multiplying them in a laboratory. Okay, and then again they're returning it to the patient, which will lead to the patient having more regulatory T cells and thus helping them to prevent the autoimmune diseases and also other uh, issues in the immune system. Okay, so there are many ways which the scientists are doing. They're modifying T cells and putting antibodies on the surfaces of this modified T cells, and then they're acting as address labels. So then they send these researchers, they're sending uh, this T cells, okay, to a transplanted liver or kidney, and then they're seeing how they can protect that organ, okay, how they protect the organ from the immune system so that it doesn't get rejected, it is accepted, all these things. So many ways the scientists are trying to use T-Rex to combat the diseases and prevent many type of diseases in the human body. Okay, so this is a very very important study, and this is one type of study which will help further to in many ways to act against many type of diseases which till now solutions have not yet been found. No solutions have been found, so it's very very helpful. So let's see the future what else we get, what else way they can be used. So this is all about in today's video. So do not forget to check the immunity video the link is given above. So please click on the link and you can watch the immunity class. And if you like the video, you click on the like icon, and also do not forget to subscribe my channel for more videos and share this with your friends. Thank you so much.